Hello, students. Today I'm going to tell you about antibodies. You know that antibodies, they are glycoproteins and they are produced by the B cells. The B cell has got a receptor on its surface, which is called the B cell receptor or BCR. And this BCR actually is an antibody attached to the B cell. And this antibody, uh, you know, it it recognizes antigens very specifically. One B cell has got a receptor that is specified for only one type of antigen. Similar is the case with T cell as well. So when it comes in contact with an antigen, you know that B cell transformation takes place. It becomes a plasma cell, and the plasma cell then secretes the same BCR, B cell receptor, or the same antibody which is also called immunoglobulin. We call it immunoglobulin or antibody. These are actually glycoproteins. We call them glycoproteins because different types of carbohydrates that you see here are attached to these antibodies or these proteins. That's why we call them glycoproteins. On average, you can say that antibody is a molecule of 150 kilodalton, and it has, um, uh, a light chain which is indicated here by this yellow color, and it has, a heavy chain which is shown here by the green color. The light chain, the yellow, it's about, molecule of 25 kilodalton, and the heavy chain is about 50 kilodalton, and as a whole, the antibody is about 150 kilodalton. And just like any other protein, it has an N terminus here and it has a carboxy terminus here. And towards the N terminus there, this is the antigen binding site, which is known as FAB, F-A-B, A-B for antigen binding and F for fragment. So this is the fragment, antigen binding fragment, which is known as the FAB region. The antigen binds here on this Y-shaped structure that you see here, and this region, which is shown here, which solely is consisting of the heavy chain. This is the FC region. This is the crystallizable fragment. That's why it is known as the FC region. And this FC region has got receptors for, its attachment on different, you can say, immune cells, just like the mast cell, it has got an FCR or receptor for the FC region of the antibody. So it can attach with different types of immune cells, and the process of opsonization that on phagocytes also, you can find this receptor where the antibody binds. On one hand, it would bind with the antigen, just like this one. On the other hand, it would bind with immune cell. So the process of phagocytosis is speeded up, and that speeded up form of phagocytosis is known as opsonization. Here you can see the structure is more detailed, where you find this amino group here and the carboxy group, which is here. And you also see that all these fragments or the segments, they are attached to each other by disulfide linkages that are shown here. CHO indicates the attachment of carbohydrates. And the hinge region also here that you see here is linked with each other by the disulfide linkages. Both the heavy chain and the light chains have these variable regions. This one, and these variable regions are responsible for attachment of the antigens. And there are different isotypes of immunoglobulins, which are 5 different types of antibodies that are shown here, which is IgG, IgM, IgD, IgA, and IgE. These are 5 different classes of antibodies. And the classes of antibodies, they differ from each other because of these heavy chains that they have, which is gamma, for instance. In this case, in the case of IgG, it is mu. In the case of IgM, it is delta. In the case of IgD, it is alpha in the case of IgA and epsilon in the case of IgE. So all the antibodies, they differ from each other because of these long chains that, the heavy chains that they have. And if you look at the different classes of antibodies, you would find some of the antibodies that are similar to each other than others. For instance, IgM and IgG are structurally, similar to each other because they do not have the hinge region shown in other antibodies right here. You see, they don't have a neck here. They are neckless. These two antibodies are neckless. IgM and IgE, these are necklace antibodies. And if you look at, the segments of the heavy chains, they have more segments of the heavy chain here. So this V here indicates the variable and C indicates the constant region. And antibodies have constant region of the heavy chain 1, constant region of the heavy chain 2, constant region of the heavy chain 3. Here you can see that constant region 1, 2, 3, and 4. 4 are found in case of IgM and 4 in case of IgG. And V H means variable region of the heavy chain. This is given here or here, and this is also of the other heavy chain. So you can say there is a single variable region of the heavy chain and 4 different, uh, uh, constant region chains. In other antibodies, you can see that there are CH1, CH2, and CH3, 3 segments of the heavy chain. And there also is variable region given here, at the top. These antibodies also not only, they are different from each other in their hinge region that you see here. Some of the antibodies, they are secreted as monomers, and some are secreted as pentamers. Some are secreted as both monomer and dimer. IgA is secreted as monomer and a dimer. And IgM, which is the first antibody to be secreted when there is some kind of infection, in that case, this particular antibody, IgM, is secreted. So IgM, when it is secreted as a pentamer, then this pentamer would, make such a star-like, appearance. And that actually ensures that, 1, 2, 3, 4, and this is the fifth antibody. So this kind of structure that will be made here. And this is for to increase the chances of binding more antigens because this antibody is not very flexible as compared to the others which have the neck region or the hinge region. This antibody doesn't have that. So to maximize the chances of antigen binding on its various sites or to engage more antigens at the same time, this antibody, which is first of all secreted by the B cells when there's some infection, IgM, is secreted in the form of a pentamer. And then the B cell would shift from IgM to IgG, which is known as class switching. And why it happens with the passage of time, the B cell would refine this particular IgM antibodies. This was made in a hurry situation. And now the B cell would have, plenty of time as the infection would progress from acute phase to, the chronic phase. So the B cell also gets ample time to refine or do some fine tuning to this antibody that it is producing right here or secreting right here. So it would do the fine tuning of IgM and convert it into IgG. Different types of mutations takes place, somatic hypermutation. You would have heard different terms, that the B cell implies these strategies here to introduce such kind of mutations in the antibodies coding genes in order to make a high affinity antibody. So the purpose of this class switching is to make a high affinity antibody, which can bind with the antigen with more strength. And that's the whole purpose of it, to do the class switching of the antibodies. What are the antibodies used for? The antibodies are used for neutralization of the antigens directly to render them incapable of doing any harm to the body, or they can label the antigens and target them for the phagocytes to engulf them. So that's all about the antibodies lecture. More detail will be shared later. Thank you.